Hello everyone, my name is Kaelyn Fogelman and today I will be presenting my dissertation research titled The Feeding Ecology and Lethal Thermal Tolerances Among Selected North American Freshwater Mussels within the order Unionida. My advisor is Dr. Jim Steckel and my committee members are Dr. Brian Helms from Troy University, Dr. Hisham Abdulrahman and Dr. Rusty Wright from the School of Fisheries, and Dr. Brendan Higgins from Biosystem Engineering. Dr. Moises Bernal from the College of Science and Mathematics has graciously accepted the role of my external reader. Unionid mussels are one of the most imperiled group of organisms in North America, and the objective of my research was to identify and elucidate knowledge gaps that can aid in conservation and management efforts. I will first present a general introduction discussing the scope of the taxa within my work and the two knowledge gaps that this work focuses on, feeding ecology and thermal tolerance. When investigating feeding ecology, for Chapter A, I first evaluated food resources of unionids in a lentic system using stable isotope analysis. For Chapter B, I then performed a similar study investigating the feeding of mussels in lotic systems in central Texas using stable isotope analysis. Finally, for Chapter C, I investigated the feeding of the same individual study within Chapter B using fatty acid analysis this time. Chapter D addressed thermal tolerance, and I conducted a systematic review to summarize all existing lethal thermal tolerance data for North American unionid mussels. The unionid mussels in this study are from the North American faunal region, which extends from the Mexican Plateau through the Canadian interior and Pacific regions. In North America, there are 302 unionid mussel species, 297 of which are within the family Unionidae, and 5 are within the family Margaritiferidae. We have approximately 1/3 of global unionid mussel diversity here in North America. But unfortunately, 65% of our mussel species are imperiled. They're either classified as vulnerable, threatened, or endangered. One of the barriers that we have in unionid conservation is that we have a poor understanding of their feeding ecology. As mussels are epibenthic organisms, they are able to access both suspended and benthic food materials. While we've elucidated the general constituents of their diet, such as algae or bacteria, we are still trying to determine where these dietary components are coming from and how these animals are accessing them, either from benthic or suspended resources. Stable isotopes are a useful tool in investigating aquatic food webs. When moving up the food web, we expect a consumer to be enriched approximately 3.4 parts per thousand nitrogen above their primary food source, and the consumer to be enriched approximately 1 part per thousand carbon above their food source. Carbon signatures are able to inform us of the primary basal food source of a consumer, while nitrogen signatures inform us of trophic level. In our study, we would expect to see our mussel consumers enriched approximately 3.4 parts per thousand nitrogen and 1 part per thousand carbon above their primary identified food source. Stable isotope analysis is an effective tool in mapping aquatic food webs. But this technique does have limitations when analyzing bulk food compartments and differentiating between dietary carbon sources. Quantitative fatty acid analysis is a technique that can identify food sources and can aid in mapping aquatic food webs when coupled with stable isotope analysis. Specific fatty acids and groupings of fatty acids can be used to distinguish between algal and bacterial-derived food sources, whether food is of a toxinous or allochthonous origins, and whether individuals are suspension or deposit feeding. Additionally, ratios of fatty acids and concentrations of individual fatty acids can inform us of the quality and quantity of a consumer's food sources. The imperilment of unionid mussels is likely compounded by global climate change. Mussels are particularly vulnerable to thermal stressors because they are sedentary, which limits their ability to seek refugia from stressful temperatures. They also have a complex parasitic life cycle requiring host fishes that may have different thermal requirements. However, there is a limited understanding of their thermal ecology on which to base conservation, restoration, propagation, and management strategies. When considering effects of global climate change and increasing surface water temperature on Unionida mussels, it is imperative to synthesize current information on their thermal limits. Once this has been completed, knowledge gaps, future research directions, and management strategies can be identified. I will now discuss my first research chapter, which we have titled here Chapter A, The Feeding Ecology of 3 Freshwater Mussel Species in a North American Lentic System. This research has been submitted to hydrobiologia. In this study, we quantified stable carbon and stable nitrogen isotopic signatures for the 3 mussel species and their potential food resources within a lentic system using stable isotope analysis. Our objectives in this study were to first determine whether mussels were eating the same thing regardless of species. Second, determine whether mussels were accessing benthic or suspended food resources. Third, evaluate whether feeding changed with mussel size. And finally, compare mussels that had been immersed and mussels that had been emersed for 8 weeks. For our lentic system, we looked at Gant Lake in southern Alabama, and we sampled 3 species there: Elliptiopullata, Fusconea escambia, and Uterbacchiana hartfieldorum. Elliptiopullata is a common species, Fusconea escambia is a federally listed species, and Uterbacchiana hartfieldorum is a species of concern. These species were all sampled immediately following a scheduled drawdown event of Gant Lake. Elliptiopullata was sampled immediately after the drawdown, but was also collected alive and sampled after being immersed for 8 weeks after the lake drawdown. We sampled mussel foot tissue sublethally during the drawdown event. Prior to the drawdown, we sampled CPOM, or coarse particulate organic matter associated with decaying leaves or detritus from the littoral zone. We then sampled FPOM, or fine particulate organic matter associated with benthic sediments from both the littoral and limnetic zones, and suspended particulate organic matter isolated from the water in both the littoral and limnetic zones. After returning samples to Auburn University, FPOM and SPOM samples were filtered to isolate particulate material. All of our samples, the food sources, and the mussel foot tissue were then dried to a constant weight and encapsulated before being sent for analysis of stable nitrogen and carbon isotopes using GCMS at Washington State Stable Isotope Core Laboratory. We'll now look at the results for our first question, which was, are mussels eating the same thing regardless of species? Here we have a biplot of the isotopic signatures of our food sources and mussel consumers with stable carbon isotope on the x-axis and stable nitrogen isotope on the y-axis. There was no significant difference across recently immersed species in nitrogen-15 signatures, but there were significant differences in carbon-13 and carbon-nitrogen ratios between Fusconea escambia and recently immersed Elliptiopullata. Fusconea escambia was more carbon depleted than recently immersed E. pullata, and Fusconea escambia also exhibited a significantly lower carbon-nitrogen ratio relative to recently immersed Elliptiopullata. Uterbacchiana hartfieldorum exhibited intermediate carbon and carbon-nitrogen ratios and was not significantly different from either of the other species. When addressing the question of whether food resources were of benthic or suspended origins, for all of our species, limnetic FPOM was the dominant carbon source, with littoral FPOM, limnetic and littoral SPOM, and CPOM comprising relatively minor contributions. limnetic F-palm dietary contributions to all species were greater than or equal to 98.7%, and contributions from all other sources were less than 1%. When looking at the relationship between isotopic signatures and mussel length, we saw isotopic signatures have a significant relationship for only one species, Uterbacchiana hartfieldorum. As shell length increased, carbon signatures decreased, and nitrogen signatures increased for this species. Finally, when looking at differences between Elliptiopelata that was sampled immediately after the drawdown event and was then sampled again after an 8-week immersion period, we found that 8-week immersed Elliptiopelata were significantly nitrogen enriched relative to recently immersed Elliptiopelata. So in conclusion, when addressing variation across species, we found that mussel isotopic signatures had minimal variation across species. For all species sampled, Bayesian mixing models suggested carbon derived from limnetic benthic fine particulate organic matter, or FPOM, contributed on average 99% to the mussel diet. A central question when developing conservation plans for mussels is whether all species in a community have similar environmental needs or whether they need to be managed differently. In this study, the answer was unequivocal with regard to food sources. All species showed evidence of using FPOM as a major food resource, indicating that the management of benthic food resource quality would benefit multiple species. For our second question of whether our food sources were benthic or suspended, 99% of our dietary carbon came from a benthic source, and less than 1% of contributions came from a suspended source. This provides evidence that the benthos can be exploited as food sources by mussels, and we should consider the quality of benthic food sources available when developing management strategies for mussels. When looking at isotope and body size relationships, we detected a negative relationship between shell size and carbon depletion and a positive relationship between shell size and nitrogen enrichment for Uterbacchiana hartfieldorum. Uterbacchiana hartfieldorum sampled in this study fell into two size classes, which were 40 to 50 millimeters and 80 to 100 millimeters. This pattern in length versus isotopic enrichment or depletion may be due to diet shifts associated with observed ontogenetic niche shifts in habitat use between young individuals, which tended to be found in shallower habitats, and older individuals, which tended to be found in deeper habitats. Elliptiopelata that were collected live but immersed for 8 weeks after the reservoir drawdown event were nitrogen enriched compared to Elliptiopelata that had been collected immediately during the drawdown event. A mechanism that causes nitrogen enrichment in consumers is self-catabolism or self-trophic enrichment, which consumers can undergo when they are experiencing nutritional or environmental stress. Because immersed individuals had significantly higher nitrogen signatures than immersed individuals, this provides evidence that they were catabolizing their own tissues during immersion. What we ultimately see here is that stranded unionids may rely on internal energy sources to survive periods of desiccation. We'll now move on to my second research chapter, which is titled here Chapter B, The Relative Importance of Suspended versus Benthic Food Resources to Freshwater Mussels in Central Texas, USA. This research has been published in Freshwater Biology. In this study, we quantified stable carbon and stable nitrogen isotopic signatures for 5 mussel species and their potential food resources across 4 watersheds and 3 seasons in Central Texas, USA. Our objectives in this study were first, to determine if mussels were eating the same thing regardless of species, location, or season. Second, determine whether mussels were using benthic or suspended food resources. And finally, evaluate whether mussel feeding changed with size. In Texas, we sampled mussels and food resources in 4 rivers. In the Llano River, we looked at Lampsillus bracteata and Cyclonius petrina, which are both state-listed Texas endemics. In the Navasota River, we sampled Cyclonius pustulosa, which is a widespread common species in North America. In the Colorado River, we looked at Cyclonius petrina, which is our Texas endemic, and Cyclonius pustulosa, which is our widespread species. And then finally, in the Guadalupe River, we looked at Cyclonius neckii and Lampsillus bergmani, which are both newly described species in Texas that are highly endemic to the Guadalupe River basin. Sampling occurred seasonally in spring, summer, and autumn of 2017. At each of our sites, we sampled mussel foot tissue. We then sampled SPOM, or suspended particulate organic matter isolated from the water column, FPOM, or fine particulate organic matter associated with benthic sediments, and CPOM, or coarse particulate organic matter associated with decaying leaves or detritus. All of our samples, including our food sources and our mussel foot tissue, were processed and analyzed identically to the samples in Chapter A. When addressing our first question of whether mussels are eating the same thing regardless of species, location, or season, we found that when controlling for the effect of species and site, mussels had the most depleted carbon signatures in spring and the highest carbon signatures in summer. Mussels were more nitrogen enriched in spring and summer than in the fall season. When sampling the same species across sites, we did find intraspecific differences. Cyclonius petrina was more carbon depleted and nitrogen enriched in the Colorado River compared to the Llano River. Cyclonius pustulosa was more nitrogen enriched in the Colorado River compared to the Brazos River. When more than one species was sampled within a site, we saw minimal interspecific differences, with our mussels having almost identical isotopic signatures. When looking at variation in carbon and nitrogen ratios, we saw significant variation in seasonal ratios. Carbon-nitrogen ratios were higher in summer than in fall and spring for all species except for Cyclonius neckii. Summer ratios were 2.6 to 3.4 times higher in the summer than in spring and autumn for 4 of our 5 species. When looking at whether our food sources were of benthic or suspended origins, we found that in the Llano River, our 2 species had co-contributions from CPOM, which was our detrital material, and SPOM, which was our suspended material. In the Navasota River, Cyclonius pustulosa received contributions from CPOM, and in the Colorado River, our 2 species received contributions from CPOM and SPOM. Finally, in the Guadalupe River, Cyclonius neckii predominantly used CPOM, or a detrital material, and bergmani predominantly used SPOM, or a suspended material. Overall, our species utilized CPOM with secondary contributions from SPOM. When looking at the relationship between feeding and age or size, we found positive relationships between mussel shell length and carbon and nitrogen isotopic signatures. Shell length was positively related to carbon and nitrogen for Cyclonius neckii, Cyclonius petrina, Cyclonius pustulosa, and Lampsillus bergmani, but not for Lampsillus bracteata. When looking at our first question of whether mussels were eating the same thing regardless of species, location, or season, we found that isotopic signatures and carbon-nitrogen ratios were consistent across species within a site. There was variation across sites, but variation in consumers was minimal within a site. Additionally, primary food resources were similar across all sites, with the predominant food sources being CPOM, or a detrital material, and SPOM, or suspended material. This suggests that there are site-specific as opposed to species-specific drivers of feeding. While not a specific objective of our study, we did make an observation that our consumers consistently had high nitrogen isotopic signatures relative to their food sources, and we also saw high carbon-nitrogen ratios in the summer season, so we investigated the mechanism behind this. Typically, the nitrogen isotopic signature of a consumer is enriched approximately 3.4 parts per thousand above its primary food resource. The value of 3.4 parts per thousand nitrogen is a fractionation from one trophic level to the next. The observed nitrogen stable isotopic signature in our muscle tissue was enriched by 5.2 to 8.7 parts per thousand above their respective predominant food resource, either CPOM or SPOM, for all of our species and sites except for Lampsillus bracteata in the Llano River. Nitrogen enrichment above one trophic level, traditionally 3.4 parts per thousand, can be caused by self-trophic fractionation due to catabolism of tissues that leads to metabolic retention of nitrogen stable isotopes in the organism. This increased nitrogen enrichment has been shown in oysters and fish and is indicative of nutrient or environmental stress. We saw a sharp increase in July carbon-nitrogen ratios relative to April and October, suggesting nutrient and environmental stress were particularly high in the summer. Taken together, our results suggest that the co-dominant CPOM and SPOM resources may not have been of sufficient quality or quantity, and most species were subjected to thermal stress and food limitation in July. Across all species and sites, Bayesian mixing models suggest carbon derived from coarse particulate organic matter, or CPOM, contributed on average 51% to the mussel diet, and carbon derived from suspended particulate organic matter, or SPOM, contributed 41% on average to the mussel diet. Mussels were potentially consuming microbial biofilms associated with CPOM pools, but preferential assimilation of detrital biofilms remains to be quantified. Together, these data suggest that carbon in mussel tissues was of benthic and suspended origins. These findings provide further evidence that mussels effectively exploit benthic in addition to planktonic food resources, which emphasizes the need to maintain riparian habitats and allochthonous inputs. Most of our species had a significant positive relationship between isotopic ratios and shell length, which indicates changes in food resource incorporation with increasing mussel size. I will now discuss my third research chapter, titled here Chapter C, which is evaluating the spatial and temporal fatty acid variants to inform food resources of unionid mussels. Our objectives in this study were to quantify food resources for 5 species of unionids in 4 lotic systems in Central Texas using fatty acid analysis. This study used the same mussels that were used in Chapter B. Our specific objectives were first to quantify fatty acid profiles present in the mussel foot tissue. Second, evaluate whether fatty acid profiles were the same across species, location, or season. And third, determine what fatty acids can tell us about the mussel diet. Stable isotopes in Chapter B were able to tell us where food resources were coming from for mussel consumers, which we identified they were from coarse particulate organic matter associated with benthic detritus and suspended particulate organic matter isolated from the water column. What fatty acids can potentially tell us is what within those bulk resource pools mussels are utilizing as food sources. For this study, I used mussel foot tissue from the same individuals used in Chapter B. I first extracted and transesterified the lipids present in the mussel foot tissue, which were then isolated and analyzed using GCMS. For each mussel, I identified the individual fatty acids present in the tissue and calculated the concentration of each fatty acid relative to a standard. When addressing our first two objectives, to first quantify fatty acid profiles and determine variation across season, drainage, and species, of the 37 fatty acids that I analyzed for, 22 of them were present in mussel foot tissue. There was variation in fatty acid profiles present in the mussel foot tissue, which I saw across species, site, and season. While there were differences in the profiles across species, site, and season, it is important to note that our R-squared value, or the measure of differences across profiles, was very low, and 55% of fatty acids were common across all mussels. When addressing our third objective of what fatty acids can tell us about the mussel diet, we were able to make inferences about mussel food quantity and quality. When evaluating food quantity using the concentration of fatty acids in mussel foot tissue, we found that quantity was highest in the Colorado River and in the summer season, and quantity was lowest in the Navasota River and the fall season. Food quality was determined using ratios of fatty acids, and it was highest in the Guadalupe River, the summer season, and in Cyclonius neckii. Food quality was the lowest within the Colorado River, in the fall season, and for Cyclonius pustulosa. Based on the ratios of omega-3 to omega-6 fatty acids, we were able to determine that our consumers were both deposit and suspension feeding. Consumers that exclusively use deposit feeding have low omega-3 to omega-6 ratios of approximately 0.15 to 0.22, and suspension feeders have comparatively high ratios of greater than 5. Our mussels had intermediate signatures of 1.5 to 3.8, which is seen in aquatic vertebrates that are using both suspension and deposit feeding modes of feeding. 90% of the fatty acids found in our mussels were not source specific, but 10% of the fatty acids are dietary tracers that are unique to either bacteria or algae. Our algae-specific fatty acids were 10 times more abundant than our bacterial-derived fatty acids in mussel foot tissue. Comparing these results with what was found in Chapter B for these same individuals, there is support that mussels are using both S-palm and C-palm as their predominant food sources and accessing them with suspension and deposit feeding. Within these bulk resource pools, fatty acids indicate that algae is a primary dietary food source. I will now discuss my fourth and final research chapter, which I've titled Chapter D, a review of North American freshwater mussel lethal thermal tolerance. Our objective for this study was to synthesize all current information on Unionid lethal thermal tolerance limits. My specific objectives for this study was to first summarize lethal tolerance data by life stage and taxa. Second, put this data within an ecological and global change context. And finally, identify future needs for evaluating Unionid thermal tolerance. For this study, I conducted a systematic literature search. I included studies which focused on Unionid mussels within North America and included lethal thermal tolerance data. I excluded papers which looked at other bivalves such as jersinid mussels or spherid clams, studies that evaluated only sublethal stressors, or studies that evaluated coupled stressors of both temperature and another contaminant. I found 71 papers, 13 of which were included. Studies were categorized first by life stage, Glaucidia, juvenile, or adult. They were then categorized by either static or dynamic exposure methods. Static studies are those in which an animal was acclimated at a specific temperature and then exposed at a different but stable temperature. Dynamic studies are those in which the temperature that the animal is exposed to changes over time, which can include critical thermal maximum or ramping studies, and studies where animals are exposed to ambient or diel temperature fluctuations. Within my static studies, I classified them into exposure durations, which were acute, medium, or chronic exposures. And then for all of my studies, the data was then organized by taxa. The data was organized in this way so that when we had a paucity of data, such as when we did not know the lethal thermal tolerance for a species, this data is able to be used to compare congeners or to compare within a life stage. When addressing our first objective of summarizing our data by life stage and taxa, our literature search revealed that for the family Unionidae, there is data existing for only 28 of 297 species for at least one life stage. And for the family Margaritiferae, there is lethal thermal tolerance data for 0 of the 5 species in North America. So in total, only 28 of 302 North American species are represented for at least one life stage with existing lethal thermal tolerance data. This is 10% of the mussels within North America. This is an example of the way that the data was represented in this manuscript. So on the left side here, we have all of our static lethal thermal tolerance data organized by taxa and life stage. So this includes exposure durations of acute or medium and chronic. And then the graph on the right, this is our dynamic studies, which in this case only had adult data represented. So here I have a few things that I would like to point out when we summarize all of the data together. The first thing that I would like to point out is the number of species that have been evaluated for our test types when looking at life stage. So for juveniles, there have only been 3 species evaluated for static, medium, and chronic exposure lengths. For adults, there have only been 4 species looked at for static acute studies, 3 species for static, medium, and chronic studies, and 4 species for dynamic critical thermal maximum studies. When looking at the average lethal thermal tolerance limits for mussels, the lower range is 30.1 degrees Celsius, which is found for juveniles, and the upper lethal thermal tolerance value is 39.5 degrees Celsius, and that is for adult dynamic critical thermal maximum studies. Here I have a graph representing the true takeaway from this literature review. The main takeaway from this study is the true paucity of data that we have. So across test types within life stages, the gray represents the number of species that have been evaluated, and the dark blue represents the number of species that have yet to be evaluated. So this is a good graphical representation of what data we do have and what data we have yet to collect. When summarizing all of this data, we found that across life stage, the mean LT50 or median lethal temperature at which 50% of exposed individuals will experience mortality was 32.8 degrees Celsius for Glaucidia. The mean LT50 for juveniles was 35 degrees Celsius, and the mean LT50 for adults was 35.9 degrees Celsius. We generally found that Glaucidia were less tolerant than juveniles or adults of the same species, but this was not always the case. There were some species evaluated where Glaucidia was more tolerant than older life stages, but again, there were few comparisons across life stages for a species. So this observation has to be taken with a grain of salt, as we have very few conclusions to make this inference off of. Looking at the thermal sensitivity of tribes for acute LT50s, we found that Amblyomini was the most thermally tolerant tribe with an average LT50 of 35.6 degrees Celsius, and Quadrillini was the least thermally tolerant tribe with an LT50 average of 33.6 degrees Celsius. For medium or chronic LT50s, we again found that Amblyomini was the most thermally tolerant tribe with a mean LT50 of 36.7 degrees Celsius, and Lampsilini was the least thermally tolerant tribe with a mean LT50 of 33.6 degrees Celsius. These observations, again, have to be tempered because for Amblyomini and Quadrillini, which were the two most thermally tolerant tribes, there was only one species evaluated within these tribes, within one study, within one geographical location, and the species studied do have widespread distribution within North America. Putting this data within a climate change context, if we look at the grand mean for acute LT50s across all life stages, this value is 34.6 degrees Celsius. So again, the LT50 is the median lethal temperature at which 50% of individuals will experience mortality. The chronic LT50 was 32 degrees Celsius, with the chronic LT50s generally being lower than the acute LT50s. One of the takeaways from this is that faunal regions that have high degrees of endemism, such as the Pacific Northwest or the Atlantic Northeast, are predicted to have greater rates of warming in the near future. Within these regions, there were few endemic species that were represented within this review. There were no species from the Pacific Northwest that we have thermal tolerance data for, and only a few within the Atlantic Northeast. The southern faunal regions, which we do have slightly more data on mussels for, have summer temps that can already reach known LT50s for mussel species that have been evaluated. This review only looks at lethal thermal tolerance limits, but we also need to investigate sublethal effects because sublethal effects can and will occur at lower temperatures than the lethal thermal tolerance limits. So for our overall conclusions and future directions based on what was revealed within this literature search, one of the negative findings was that we still have yet to determine thermal tolerance data for 90% of North American species. One of the positive takeaways from this study was that acclimation temperatures had minimal effect on lethal thermal tolerance endpoints for species that were evaluated. Acclimation temperature is something that is very important for fish, and acclimation temperature can have a significant effect on lethal tolerance endpoints. But for mussels, we have found that this may not necessarily be the case because for only 36% of studies was there an effect on acclimation temperature. So potentially in the future, what we can do instead of looking at multiple acclimation temperatures for a single species, instead we can use one acclimation temperature and instead focus on more species or subpopulations of species across different latitudinal gradients. One thing that we do need to investigate is the lethal thermal tolerance limits for mussels within the Northeast Atlantic and the Pacific Northwest, where we have high degrees of endemism and we are predicted to have high impacts from warming surface water temperatures. To summarize the findings from all of these studies together, for Chapter A, we looked at 3 Unionida mussel species within one lentic system in just one season using stable isotope analysis, and we found that fine particulate organic matter associated with benthic sediments was their primary food source. For Chapter B and C, we looked at 5 species of mussels across 4 lotic systems in 3 seasons using both stable isotope and fatty acid analysis. And this study found that coarse particulate organic matter associated with benthic sediments and suspended particulate organic matter associated with particulate matter in the water column were their primary bulk food sources, but within those bulk food sources, algae was the dominant dietary item. Some of the findings that are consistent across these three studies were that we saw minimal species variation, suggesting that we're able to utilize a community-wide management approach when it comes to feeding ecology. An important takeaway is the importance of benthos as a food resource for mussels. And finally, we saw elevated nitrogen or carbon nitrogen signatures for some of our mussels, which reveals evidence of self-catabolism that mussels may undergo as a coping mechanism for periods of desiccation or as a response to nutritional or environmental stress. In our final chapter, I synthesized all known lethal thermal tolerance data for unionid mussels in North America. And what we found is that only 10% of species have known upper lethal thermal tolerance limits. The median lethal temperature at which 50% of individuals will experience mortality ranged from 21.4 to 44.4 degrees Celsius, with a grand mean of 34.2 degrees Celsius. One of our findings was that faunal regions that are most threatened by climate change, such as the Pacific Northwest and the Atlantic Northeast, have high degrees of mussel endemism, but little known thermal tolerance data for those mussels. One of our major takeaways is that instead of focusing on mussels at different acclimation temperatures to evaluate lethal thermal tolerance limits, instead we can focus on additional species and additional subpopulations of species across latitudinal gradients, as acclimation temperatures may not have as great of an impact on thermal tolerance endpoints as it does for other aquatic ectotherms. I would like to thank everyone who made this research project possible. There were multiple studies that were conducted over the course of 5 years. From my funding sources in Texas and in Alabama, and from the federal agencies, to all of the lab members that are currently here and have already graduated that assisted in these projects, and finally, everybody from agencies that assisted in making fieldwork possible. This truly was a labor of love, and it would not have been able to be done without the assistance of countless people who are mentioned here and others that are too numerous to mention. And thank you everyone for attending my virtual exit seminar. If you have any questions about any of the research that I've conducted or any of the findings, you can find some of these manuscripts already published, or you can email me with questions, and my email is at the bottom of the slide here.